All right, sorry for being late logging in. I once again ran into a problem where they wanted to verify my existence and sent me a text message. And I had to go look for my phone because I took it shopping with me. And then I just put the groceries down and then got ready for class. And when I tried logging in, like I said, I needed to verify my existence and they sent me a text message. And I have to complain about that. I think that happened just a few days ago. Um, I don't know why they're so, I don't know, demanding of making sure that I'm actually the person logging in. Anyways, are there any questions before we get started? Can somebody confirm that you can hear me?
I can hear you.
Thank you. All right, let's go ahead and get started. I think I can close that down. Let's go ahead and get to our website. I did some grading. I think it was in the lab and maybe the lecture worksheets. So take a look at that. I got a few emails I haven't looked at. All right, let's begin. Today is November 16th. Let me see, I don't think I'm sharing my screen. You should see my screen now. And we're supposed to be working on chapter 8. We don't need to finish chapter 8 until Thursday. Wait a minute, is today Thursday? Today is Thursday, isn't it? Yeah, we're supposed to finish chapter 8 today. We'll probably fall behind schedule. There is no lab today, so we'll be logging in for the first 15 minutes to answer questions, but you should be working on your unknown project. Any questions about any of that? I have not taken a
Nope.
look at your, thank you, at your infectious disease project. I'll try and get started on that Thanksgiving weekend. All right, let's begin with chapter 8. I have that we finished talking about protein translation. So let me talk about some complications of protein translation. You're actually seeing a complication of protein translation here, but you're probably not. Oops, that doesn't look good. Oh, there it went. Sorry, I've got a frog in my throat. We're actually looking at a complication of protein translation here, but it's probably not apparent to you. But there's the message, the DNA molecule right there. You can barely make it out. And then right here is a messenger RNA. And there's another messenger RNA and another messenger RNA, another, and another. And this complication is happening in both prokaryotes and eukaryotes. Here is one gene, I don't know, something like right here to about there. And as you can see, there's more than one messenger RNA being made for this gene at the same time. In fact, there's 1, 2, 3, 4, 5, 6, 7, at least 8 messenger RNAs being made at the same time. And this is a problem in both prokaryotes and eukaryotes, or I should say not a problem, a complication. And that is you can have more than one message being made at the same time. And this one is either finished or almost finished. And that one is just starting. That's why it's much smaller on this side, and it gets larger as you come this way. Any question about any of that? All right. In prokaryotes, you have another complication, and that's because in prokaryotes, the DNA is sitting in the cytoplasm. You do not have this complication in eukaryotes because their DNA is in the nucleus. And the complication is, you see these, I don't know, roundish things here? Those are ribosomes. And this is the artist picture here. There's the molecule DNA. There's the RNA polymerase, and you can make out just barely the blue line. Oops. The blue line showing you why did that happen. Showing you the messenger RNA. And then those red things on the messenger RNA are ribosomes. And you can notice that there, first of all, there are more than one ribosome on one messenger RNA. And you notice in prokaryotes that the ribosomes will bind to the messenger RNA and begin protein translation before RNA transcription has finished. So this one here, the sorry, I'm drawing a blank here for what the heck that's called. The RNA polymerase is just starting to make the messenger RNA. And ribosomes have already bound to it, in fact, two of them, and they're beginning to make the proteins. The proteins aren't shown in this picture here, and they're not showing up in the electron microscopic image either, although it could be that, like I said, the small grains right there could be, come on, mouse, could be a messenger, a protein, but it's not small, big enough to show up, at least not clearly. Well, let me see now. So the RNA polymerase will begin binding at the start of the messenger RNA. Come on, mouse. And then we'll move down the messenger RNA. And so this one here would have the largest protein, and we just can't see the proteins there. The point is, because prokaryotes have their DNA in the cytoplasm, we have RNA transcription occurring at the same time as protein translation is proceeding, meaning the two are happening at the same time. Now, in eukaryotes, you don't have that happening because the messenger RNA is made in the nucleus, and then the finalized messenger RNA will then move into the cytoplasm where protein translation will occur. But in prokaryotes, it's a complicated mess. You have, first of all, more than one messenger RNA being made on the same gene, and you have that problem in, or complication in eukaryotes too. But in prokaryotes, you have protein translation beginning before RNA transcription has finished. Any question about any of that? All right. In eukaryotes, we have another complication, and that is when the messenger RNA is first made, the initial RNA transcript, it includes regions of the DNA that we call exons, and an exon is a region of DNA that codes for the final product, which in this case, since this is a messenger RNA, that would be a protein. And it also includes the initial RNA transcript, the introns, which are regions of the DNA which do not code for proteins in a messenger RNA. this initial RNA transcript, which includes introns, cannot leave the nucleus, meaning the nuclear membrane pore has a gateway, and any messenger RNA that still has the introns in it is not allowed out of the nucleus. It cannot be allowed out of the nucleus until the introns are removed and the exons are put together. Now, the term that's used is the introns are spliced out of the initial RNA transcript, and then the exons are spliced together to make the final messenger RNA. The final messenger RNA can then leave the nucleus, go into the cytoplasm, where it will be translated. Any question about any of that? All right. Let's briefly talk about eukaryotic DNA. It encodes coding regions, meaning DNA that actually codes for the gene product, and if it's a protein, the exons code for proteins. The introns are a region of a gene that does not code for protein. And I'm not entirely sure what introns do. They might have a role in the folding of the DNA in a chromosome. And then in eukaryotes, we have a lot of the DNA, which is not a gene. So here's one gene here, and that's the DNA. This DNA moves this way and that way. And this region of the DNA right here is non-coding DNA. And then the next gene could be down here someplace. The non-coding DNA is important in the folding of the DNA to make the chromosome. Any question about that? And the non-coding DNA, exactly what else it might be doing, we don't know. Or at least I don't know. It wasn't known when I studied genetics, which admittedly was a long time ago, but people called it junk DNA. It's not just junk DNA. Like I said, one role it does play is it helps the chromosome fold up and then become a tightly compact chromosome. If you don't recall, the DNA most of the time is unwound and it's just floating around either the nucleoplasm or the cytoplasm, and it's suspended in that plasm, meaning in mostly the liquid of the plasm. However, when a cell goes through mitosis or meiosis, it will supercoil into the chromosome, which is what you always see when you see like the X chromosome. That's the supercoiled chromosome. And that will precipitate out of the plasm, either nuclear plasm or the cytoplasm. Any question about any of that? All right. So we talked about the structure and function of the genetic material, the flow of genetic information. We talked about DNA replication, RNA transcription, and protein translation. Now let's turn to talking about the regulation of bacterial gene expression. And it's very similar to the gene expression in eukaryotes, but we're not going to talk about that because this class is mostly microbiology. We will talk about the operon model, what an operon is and how it controls gene expression. Then we'll talk about mutation, and then move on to genetic transfer and recombination. And then lastly, we'll talk about genes and evolution. All right. When bacteria cells want to regulate what they have in them, one way to regulate their contents is to control gene expression. There are other ways to control their contents, like they control the enzyme. They can inactivate the enzyme, and then no more of the product of the enzymatic reaction will be made. But you could also regulate the gene, like turn the gene off or turn the gene on. And that's one way we can regulate gene expression. There are some genes, like constitutive enzymes, that are expressed all the time at a fixed rate. And we call these constitutive genes or housekeeping genes. They're always turned on and expressed at a fixed rate. There are other genes, such as other enzymes, that are expressed only as they are needed. And these enzymes are either repressible enzymes or inducible enzymes. A repressed enzyme is one where the gene is normally turned on, but you can turn it off. And so we say the gene is repressed. An inducible gene is a gene that is normally turned off, but when the gene product is needed, the gene will be turned on. And that explains the repressible enzymes and the inducible enzymes. They belong to a gene which is either a repressible gene or an inducible gene. As I mentioned, there are ways to regulate gene expression. We're going to talk about pre-transcriptional control, and these are repressible and inducible genes. We're going to mostly talk about inducible genes using an inducible operon. And we'll talk about the lac operon, where the gene expression is controlled before the product is made. So the gene is either turned on or turned off. And we're only going to talk about the inducible system, where the gene is normally turned off, but it can be turned on when the gene product is needed. Any question about that? And we'll talk about the lac operon in specific. It's an inducible operon. But there are repressible operons where the gene is normally turned on, but when it's not needed, the gene can be turned off. We're not going to talk about that, and you're not going to be tested on it. There's other ways to control gene expression, and that's epigenetic control. And you'll be thankful to me that I'm not going to talk about it, and you're not going to be tested on it, because epigenetic control is kind of complicated, it's kind of hairy, and it's not well understood even today. When I was first going through college, we were talking about epigenetic control, and people were waving their arms saying, well, we think this might be what's happening, but exactly what's going on, we don't really know. And people were trying to study it, and they didn't have a good understanding of it. And today, they still don't have a good understanding of it. And then there's other ways to control the products that are made in a cell, and those are post-transcriptional control, where the gene is transcribed, but then you either shut down translation of the gene, or you even make the end product, let's say an enzyme, but you then inactivate the enzyme. And that's post-transcriptional control. We're not going to talk about that, and you're not going to be tested on that either. So let's talk about bacterial operons. An operon contains the gene of interest, and that we call a structural gene. And it contains maybe more than one structural gene. It depends on the gene. Some only have one structural gene, but for the lac operon, we actually have 3 structural genes in that one operon. And then operons also contain controlling regions around the structural gene of interest. And we're going to look at bacterial operons, but let me mention that eukaryote operons are similar with one exception. And eukaryote operons only have one structural gene in each operon, whereas bacterial operons may have one structural gene, or they may have a couple of structural genes in the operon. If the bacteria has a couple of structural genes in the operon, all of those structural genes will be related to each other. Like in the case of the lac operon, the first structural gene is the gene that encodes for the degradation of the sugar lactose. So we can call that the lactase gene. And I forget, it has another name, but I'm just going to call it the lactase gene. And that's the Z gene of the lac operon. Any question about that? All right. The other two genes, structural genes in the lac operon, are involved in protein metabolism of the sugar lactose. The Y gene codes for the transporter protein in the membrane, cell membrane of the bacteria cell, which will pump the sugar lactose into the cell. So the Y gene encodes the protein transporter that brings the sugar lactose into the cell. And then the A gene is important in the metabolism of the sugar lactose. Lactose will be degraded into the sugar glucose. It's a dimer. The sugar lactose is a dimer. It'll be made into the sugar glucose and beta galactose by the lactase gene. And then the beta galactose needs to be converted into glucose. And the A gene helps that conversion. So the A gene is involved in the metabolism of the sugar lactose. What it does, like I said, specifically, is it helps convert beta galactose into the sugar glucose. And all sugars are generally converted into the sugar glucose, and then the sugar glucose will be sent down aerobic respiration in the normal way. Any question about that? All right, so let's talk a little bit about the promoter and the operator. And then there's one other controlling region of the operon, which isn't shown here. It's at the end of the structural gene or genes, and it's the terminator. We've already talked about the promoter. What the promoter does, it's the region of the DNA that codes for the RNA polymerase to bind to the DNA. So the RNA polymerase will bind to the DNA at the promoter. And we've talked about the terminator also. It's the region of the DNA after the gene that when the RNA polymerase moves down the DNA not the DNA polymerase, it's the RNA polymerase. When it moves down the DNA, transcribing the RNA, it will make RNA until the RNA polymerase reaches the terminator. The terminator codes for the RNA polymerase to fall off the DNA. And then that will end RNA transcription. So that's what the promoter and the terminator do. they are involved in the expression of the structural gene. And I don't know if we mentioned it before, but let me just mention it now. A structural gene is normally what people mean when they use the word gene. Like when we're talking about the gene for the gene lactase, we mean the structural gene. When you're talking about the blue-eye gene or the brown-eye gene, we're referring to the structural gene of the operon to the blue-eyed or the brown-eyed gene. And they're the same gene, they're just different alleles at the same position on the DNA. Any question about any of that? All right, let's talk about the operator. What the operator does, it's a region of the DNA that codes for the repressor protein to bind to the DNA. And when the repressor protein has bound to the operator, when RNA polymerase then binds to the promoter and then moves down the DNA, it will get stopped by the repressor protein. And then transcription of the structural gene will not happen. So the repressor protein turns off expression of the lac operon. Any question about any of that? Let me back up. You can see in the case of the lac operon that this is the operon, and we have a regulatory gene right next to the operon. It is not in the lac operon. In fact, it has its own operon, and it looks like we're only showing the structural gene, so probably the operon will be a little bit bigger than what's shown as the regulatory gene. Normally, the regulatory gene is nowhere near the operon that it regulates. In fact, in eukaryotes, normally the regulatory gene is on another chromosome than the operon that it is regulating. Now, in bacteria, almost all of them only have one chromosome, so it's on the same chromosome because there's only one, but it's not, the regulatory gene is not near the gene that it's regulating. In the case of the lac operon, the regulatory gene is right next door to the operon that it is regulating. Any question about any of that? What the regulatory gene does is it will be transcribed, making a messenger RNA, and when that messenger RNA is translated into protein, it will make the repressor protein. The purpose of the repressor protein is to bind to the operator and then shut down transcription of this gene. Any question about that? Well, here's a question for you. Why in E. coli would the lac operon normally be shut down? Anyone have a guess? It's actually quite simple. Well, what do you know about the sugar lactose? The sugar lactose. I'm sure somebody knows about the sugar lactose. Where do you find lactose?
Milk?
Yeah, dairy products. And that's about it. How often is E. coli going to be around a dairy product? And we're not talking about E. coli in somebody's gut. Let's talk about an E. coli in the soil or on an apple tree. How often is E. coli going to be seeing the sugar lactose?
Not very often.
Yeah, not very often. And because E. coli is not going to see the sugar lactose very often, E. coli makes the repressor protein and then shuts down the lac operon. And the reason is simply because E. coli, unless it's on a dairy farm, it's not likely to see a dairy product. Now, obviously, if E. coli is in somebody's gut, and if you drink and eat a lot of dairy products, that's a different story. But we're not talking about that because E. coli can be found essentially anywhere on the planet Earth. And it is true it's in people's guts. But like my gut, for example, I don't usually eat dairy products because I'm allergic to the sugar lactose. I just don't digest it, so I don't drink milk. At least not cow's milk. I might drink rice milk or something like that, but all right. So the next question is, how do we turn on the lac operon? And you'd want to turn on the lac operon whenever E. coli finds the sugar lactose. And then if the lac operon is turned on, the sugar lactose can be metabolized. And so how do we turn on the sugar, the gene for digesting lactose, meaning how do we turn on the lactose operon? Well, whenever the sugar lactose is around, the isomer of lactose is also around. And the isomer, if you remember, one is left-handed, the other is right-handed. It's the same amino acid sequence or the same, in this case, what do you call it, chemical structure. It's just one is right-handed and one is left-handed. And that's the case of allolactose and lactose. And allolactose is around whenever the sugar lactose is around, and allolactose is the inducer. What the inducer does, meaning in this case allolactose, binds to the repressor protein, and then it inactivates the repressor protein. If the repressor protein is inactivated, it cannot bind to the operator. And if there's no active repressor protein, the operator will not get bound to. And then the promoter, if it binds, not the promoter, the RNA polymerase, if it binds to the promoter, it will then move down the operator and it won't be blocked by the repressor protein. And then it will transcribe the lactose gene. So you'll get your messenger RNA making the enzyme lactase. And another name for the enzyme lactase is beta galactosidase, which I don't use that term. I use the term lactase. Okay? Any question about how we turn on the lac operon? Well, here's a question for you. What happens to the repressor protein that's already bound to the operator? Because it has to come off before the polymerase can move past the operator when the repressor protein is bound to it. So what happens here? There's one of two things that's possible. The inducer does not bind to the repressor protein when the repressor protein is bound to the operator. And it's just that the inducer can't bind to it because the protein is in the incorrect folding pattern. And so when the repressor protein is bound to the operator, it stays there. The inducer allolactose cannot bind to it and then cause it to fall off. What actually happens is the repressor protein binds to the operator in a stoichiomic reaction. And every once in a while, the repressor protein will fall off. If there's other repressor protein around, then one of the repressor proteins will bind to the operator and the lac operon will continue to be shut down. However, if allolactose is around, and then the repressor protein falls off the operator, there will be no other active repressor protein to bind to the operator, and also the repressor protein that falls off of the operator, now the inducer allolactose can bind to it and then inactivate it. And that will ensure that the lac operon is turned on. Any question about that? The lac operon is turned on whenever the sugar lactose is around, because allolactose will also be around. Oh, I went too far. Come back here. Well, what happens to the polymerase here? Is it going to be stuck here for as long as that repressor protein is bound to the operator? Anyone have a guess? It's either a yes or no question, people.
Would it stay stuck?
No, it doesn't. It makes sense that you don't want to tie up all of your RNA polymerase, and so it binds to the promoter in a stoichiomic reaction, and every once in a while, the RNA polymerase falls off. And then once it falls off, it's then free to bind to another promoter to begin transcription of that gene. It could even bind once again to the same promoter, in which case it will get stuck because there's the repressor protein bound to the operator. But like I said, the RNA polymerase binds to the DNA in a stoichiomic reaction, and every once in a while it'll fall off, which will free it to bind to another operator. Not operator, to another promoter. All right, let's watch a little video on the lac operon. There we go.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon, beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point,
that isn't quite correct, but everything else in this video is, meaning the inducer does not bind to the repressor protein when the repressor protein is bound to the operator.
RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
All right, any questions about that video? Let me see, is there anything else I want to talk about? No, I think that's it for the lac operon. Remember, oh, there's one question I do want to, one point I want to make out, and that is you can see this messenger RNA is a little different than any messenger RNA we've ever discussed before. And the difference is this messenger RNA, one messenger RNA, is making 3 proteins. We've never seen that before. In the case of the lac operon, this occurs because there's more than one structural gene in the lac operon, where 3 proteins will be made from the messenger RNA. Beta-galactosidase, which I call it lactase, permease, which is the transport protein which brings the sugar lactose into the cell, and then transacetylase, which helps convert beta-galactose to glucose. Normally, we only talk about a messenger RNA making 1 protein, and in eukaryotes, that is always the case. So what's the answer? And that is in bacteria like E. coli, we have a gene for lactase, a gene for permease, and a gene for transacetylase. In humans or other eukaryotes, we don't have a lac operon where there's 3 structural genes on it. We certainly have a gene for Z, which is the structural gene is the lactase enzyme. We certainly have that. But do we have a permease and a transacetylase? Anyone have a guess? Nobody's going to guess? Well, the simple answer is yes. And in eukaryotes, instead of having 1 lac operon with 3 structural genes, in eukaryotes, we have 3 lac operons, 1 coding for the enzyme lactase, 1 coding for the, not enzyme, but transport protein permease, and then 1 coding for the transacetylase. So in eukaryotes, we have 3 different operons because each has only 1 structural gene in it. Any question about any of that? All right, that's it for the lac operon, the control of bacterial gene expression. Let me just mention that eukaryotes have a similar way to control their gene expression, and we're talking about an inducible gene expression, as bacteria. The only difference is that eukaryotes have more regulatory control over the operon, meaning they may have more than one regulatory gene. They may have more regulatory units controlling the expression of the structural gene of interest. But they do have a similar operon that works similar. The only real difference is they only have one structural gene to each operon. In bacteria, some of the operons have only one structural gene, and some of them have more than one structural gene in the operon. Remember, if they have more than one structural gene in the operon, all of those structural genes are related to each other, like in the case of the lac operon. All right, let's move and talk a little bit about mutation. Mutation is a change in the genetic material. So you get a change in the sequence of the DNA, that's a mutation. Mutations may be neutral, beneficial, or harmful to the individual containing the mutation. Let me talk just a little bit about a neutral mutation. And a neutral mutation is one that has no effect on the individual. Today, if we look, for example, at the blue-eyed allele and the brown-eyed allele, the change is neutral. So if a mutation were to happen where a blue-eyed individual were to mutate, I should say fertilized cell, by the way, were to mutate and become a brown-eyed individual, or vice versa, it would be a neutral mutation. But there is no selective advantage for having blue eyes or brown eyes. Now, in the past, that may not have been the case because, at least in Northern Europe, where the blue-eyed mutation originated, it looks like the blue-eyed allele, well, I'll just word it this way, the blue-eyed allele increased in frequency because starting out at close to 0% frequency in Northern Europe, the blue-eyed allele is the most common allele. So about 10,000 years ago, the blue-eyed allele may have had a selective advantage, which today it no longer has. And then another possibility is the blue-eyed allele could have been linked to a gene that had a selective advantage, and then the blue-eyed allele itself was neutral as it is today. And then it increased in frequency just because it was closely positioned next to a gene that was being selected for. You can have a beneficial mutation, and the best example that I know of happens in somebody's fruit fly lab, and they were growing fruit flies in a glass bottle. This is plastic, and it has a little cap to it. But in reality, they weren't using caps. They were using a cotton plug. There's a cotton swab, and you stuck it in there. And then the guy had food in the bottom. And then the fruit flies were in there, and they were eating the food and laying their eggs in the food. And then the fruit flies the larva would crawl up to the side of the bottle and, what is that called? Pupae. And then they would go through metamorphosis and change from the pupae stage of the fruit fly to the adult stage, which is the flying fruit fly. Okay. Well, this guy was pretty lazy, and what he would do is just put food in and the flies in, and then cap it, and then leave it. And there would be so many pupa on the side of the bottle that there was not room for other larvae to crawl up and become a pupa. What you do is your pupa, you attach the pupa to the side of the glass, and then you go through metamorphosis. Well, the side of the glass was so full of pupa that the larva could find no place to attach and go through pupation or become a pupa. We'll just word it that way. And what happened in the fruit fly lab, there was a mutation where the fruit fly could actually climb up to the bottom of the cotton swab and then form a pupa on the cotton swab. And because the guy was so lazy, just didn't change the bottles very much, the entire side of the bottle was full, and then the pupa could not go through the stage of pupa. And that made an extreme selective pressure for that mutation, favoring that mutation. And then that mutation increased in frequency in the population of fruit flies in this individual's lab. So that's the case of a beneficial mutation. And to tell you the truth, that's about the only one I know off the top of my head, which is clearly a beneficial mutation. And then there are mutations which are harmful mutations, and I'm sure you know about some of them. I'm going to mention just one, the sickle cell anemia, that if an individual has 2 sickle cell anemia genes, they, even today, tend to die before reaching adulthood. And that's because their cells, sickle cell, and the sickle cell anemia has other problems which deteriorate the body. And like I said, these individuals, even today with modern medication, very rarely reach adulthood. Any question about any of that? A neutral mutation, a beneficial mutation, and a harmful mutation. Most mutations are either harmful or neutral. And we'll talk about why so many mutations are neutral in just a minute. Or in just a few minutes. A mutagen is an agent that increases the mutation rate. You always have a background mutation rate, and that we call spontaneous mutations, mutations that occur in the absence of a mutagen. Whenever DNA polymerase is copying DNA, if ever it makes a mistake, that would be a spontaneous mutation. And then a mutagen increases the mutation rate higher than the rate of spontaneous mutation. And a well-known mutagen that many humans are exposed to is cigarette smoke. But there are others, and we try and limit the exposure to these other mutagens. Cigarette smoke, well, somebody who smokes, it's just what you're going to live with. All right, let's talk about the different types of mutations. There are 3 basic types of mutation. There's a base substitution, also called a point mutation, where one nucleotide base is changed for another base. Why it's called base substitution. And that can cause a silent mutation, a missense mutation, or a nonsense mutation. And we'll talk more about those in the upcoming slides. There can also be another type of mutation, and that is a frame shift mutation. This is where you add or pull out a nucleotide from the DNA. So instead of having 3 nucleotides, you add 1, and now there's 4 nucleotides, or you remove 1, and that means there's only 2 where there used to be 3. This is called a frame shift mutation because this mutation changes, obviously, that codon, and it changes all later codons because when you add or remove a base, you change the reading of the codons. So that codon is changed, and then all later codons are changed, at least in that gene. Any question about any of that? All right. There's also a third type of mutation called the macromutation, and that is not discussed in the book. So you have to pay attention to my lecture to understand what a macromutation is. And I don't know why the author refuses to discuss it, because there's been like 12 editions of the textbook and he's never discussed a macromutation. A macromutation is where an entire region of a chromosome is added or deleted. And if you go online and do a search for a macromutation, you'll find two searches. One is the definition that I explained, where an entire region of a chromosome is added or deleted. The other macromutation will get you confused, and I don't even want to talk about it. It's where you change a gene, and it's a controlling gene that changes the expression of many other genes. And so a lot of different genes get changed by this one mutation. And that's why they call it a macromutation. But that's not the macromutation in the DNA. I'm talking about a macromutation in the DNA where you're changing more than one base. You're changing an entire region of a chromosome. Hundreds, thousands, maybe even hundreds of thousands of nucleotides. And there's one macromutation that you probably heard of. It involves where a patient is born with an entire extra chromosome. And what is that? Anyone know?
Down syndrome?
Yeah, Down syndrome, where the patient has an extra copy of chromosome 23. Normally in humans, if a patient has an extra chromosome, that fetus will die before birth because changing that many genes at one time is just lethal. But there are some exceptions. Chromosome 23 is a very small chromosome and apparently not terribly important in the human genome, and that's why Down syndrome patients are born with it. However, they're not functioning totally normal, and that's what we call them Down syndrome patients. There is another 2 chromosomes which people can be born with, and that is there are some individuals who have an extra copy of chromosome Y, and they're XYY, and these guys, for most intents purposes, are completely normal. XYY. They are completely normal to most characteristics. The only exception is XYY individuals do tend to end up at a higher rate in prison. So you can say they're a little bit not normal, but to all other purposes, they appear to be normal. And then the other case that people can be born is XXX. These individuals tend to be very close to normal. I'm not sure what the difference is. And then you can be born with 1X, and that is close to normal, but not quite. You can notice a few differences in an individual with an X, and the individual born with an X will not be fertile. That individual will not reproduce. Although I think XXX individuals, as well as XXY individuals, can reproduce. So like I said, to my knowledge, those individuals are really close to normal. And those are all macromutations. There are some other macromutations. I don't remember it, but there's the the cat crying syndrome, where the baby, when it's born, the baby constantly cries, and they're missing an entire region of one of the chromosomes. It's not 26. It's like, I think it's chromosome 18. All right, let's talk about base substitution, where one base gets changed. Let me blow that up. So instead of putting a C here, the polymerase made a mistake, putting a T, causing a base substitution. Once this chromosome replicates, that mutation will then be fixed. And then when the polymerase sees the T, instead of putting a G there, it'll put an A, and that will fix the mutation. You can't go back to what the DNA should have been, a GC pair there, other than the rare case of a back mutation, which we're not going to talk about. So once replication happens, that mutation is fixed. Before replication happens, this mutation can be repaired. And that is the cell knows that this is the newly synthesized strand, and this is the original strand. So it knows to replace the T and put a C there. But once the DNA is replicated, that mutation becomes fixed. You can't repair it. And we're not going to talk any further about repair here. Any question about any of that? All right. So in a base substitution, we can have a silent or neutral mutation. And this silent mutation is where one base in the DNA is changed, but it does not result in an amino acid change in the protein, such as when UUU is changed to the codon UUC. That codon still codes for the amino acid phenylalanine. So there's no effect on the protein, no effect on the protein function, and this we call a silent mutation. All silent mutations are neutral mutations. And that's one reason why so many mutations can be a neutral mutation. And that's because if there are a mutation that's a silent mutation, it's a neutral mutation. Any question about any of that? All right. You can also have a missense mutation where you change one nucleotide in DNA and it results in a change in one amino acid sequence, such as when CCG is mutated to, did I do that right? CGA? No, it's supposed to be, sorry, I didn't read that right. It's supposed to be CCG. And it can be mutated to TCG, resulting in a switch in the amino acid of glycine to serine. And that we call a missense mutation, where you change a base and you change an amino acid. An example of a missense mutation is sickle cell anemia, or the blue-eye gene switching from what used to be the brown-eye gene. That is a missense mutation. You can also change one base in the DNA, and it codes for a codon that's a stop codon. That we call a nonsense mutation. where you change one nucleotide base, which changes that direct codon, so there's no longer an amino acid there, but there's no further amino acids after the stop codon. That causes a truncated protein that may change the protein so dramatically the protein cannot function. And some muscular dystrophy and some cystic fibrosis patients have a nonsense mutation. Any question about any of that? All right, so here we had a very short protein with only 4 amino acids, and the amino acid lysine was changed to the stop codon, and then we have a very truncated protein, in this case only 1 amino acid, which this is a artist's explanation because obviously there's probably no protein with only 1 amino acid in it. And also there's probably no protein with only 4 amino acids in it. All right, that's it for the base substitutions, including a neutral mutation, a missense mutation, and a nonsense mutation. You can also have a frame shift mutation where you add or remove a base from the DNA. Let me blow this up so I can see it better. Here we have the codon AAA, which codes for the amino acid phenylalanine. We remove 1A. We now have the coding of the codon AAC, which codes for the amino acid leucine. So we change this amino acid, but you'll notice we change all other codons, so all other amino acids after that change are also changed. And that's because to get this next codon, you don't just simply go AA, you take the next nucleotide, because the codon's always 3, and that's how we got the amino acid leucine. We took the next codon, which was C, coding for AAC, which codes for the amino acid leucine. Now we no longer have the codon CCG, we have the codon CGA. And you can see how a frame shift mutation changes this amino acid and all amino acids following that amino acid. And that's why it's called the frame shift mutation. Any question about any of that? Now, the simplest frame shift mutation is simply adding or subtracting 1 nucleotide. But you get a frame shift mutation if you add or subtract 2 nucleotides as well. However, if you subtract 3 nucleotides or add 3 nucleotides, you no longer get a frame shift mutation. If you add 3 nucleotides, let's say you get AAA, and you add 3 more AAAs, what you do is you add an amino acid to this protein, but it's not a frame shift mutation because the next codon would be CCG, which codes for the amino acid glycine. So after that point, there's no frame shift. The only change is we added 3 nucleotides. Okay, so you got to remember a frame shift mutation is any change where you add or decrease 1 or 2 nucleotides, or anything that's not a multiple of 3, and that would be 4 or 5, but not 6, or 7 or 8, but not 9. Any 3 multiple of 3 is not a frame shift mutation. Any question about any of that? All right, that's it for mutation. remember, you have a macro mutation of DNA, which is adding or removing an entire region of a chromosome. So let's talk about the different causes of mutations. Mutations can happen in two basic ways. They can be spontaneous, and those we call a spontaneous mutation, or they can be a mutation caused by a mutagen. And there are two types of mutagens, radiation and chemical mutagens. There are two types of radiation causing mutations. There is ionizing radiation and then non-ionizing radiation that can cause a mutation by radiation. Any question about any of that? All right. Ionizing radiation is radiation that has a lot of energy. And when that radiation hits a molecular atom, it gives the electron in that molecule of atom enough energy that that electron will leave that molecular atom. That will mean that this molecular atom will gain a -1 charge because an electron has a negative charge. And so that molecular atom, we call it an ion. And that's why it's called ionizing radiation, because this radiation creates ions. And then if those ions go and touch DNA, they will mutate the DNA. Any question about any of that? All right, so let's talk about non-ionizing radiation. A non-ionizing radiation is actually any radiation that doesn't cause ions. However, most non-ionizing radiation does not mutate DNA, like visible light does not mutate DNA. The only non-ionizing radiation that mutates DNA is UV radiation. How UV radiation mutates DNA is that UV light, when it hits DNA, it changes two thymines that are next to each other so that the thymines actually form a dimer between them. That's a dimer is two covalent bonds between two adjacent thymine. That mutates the DNA because neither DNA polymerase can move past this thymine dimer, or RNA polymerase in transcribing the DNA cannot move past the thymine dimer. And that is how UV radiation causes a mutation. I guess I forgot to mention that when you have the thymine forming a thymine dimer, the two thymines link to each other with two covalent bonds, which they should not do. And then these two thymines will stop hydrogen bonding to the A's in their corresponding strand of DNA. Any question about any of that? Cells do have a way to repair thymine dimers, but we're not going to talk about it. Let me briefly talk about chemical mutagens. We can have a chemical which also mutates the DNA. I'm only going to talk about one example, and that's nitrous acid acting as a mutagen. When nitrous acid binds to DNA, it changes this adenine, the A, such that this changed A does not act like an A. So when this DNA is replicated, the DNA polymerase doesn't read that changed A as an A. It'll read that changed A as a G. and it'll insert a C there. And then when this cell replicates, that mutation will be fixed. You can never go back other than a back mutation once you have a CG pair replacing what should have been an AT pair. And how that happened was the nitrous acid changed that A so it no longer acted like an A. It is actually a derivative of A, so it is an adenine, just not the normal one. And it's a changed A. And instead of putting a T there, the DNA polymerase will read that changed A as a G and it'll put a C there. Any question about any of that? All right, that's how nitrous acid acts as a mutagen. The spontaneous rate of mutation is for every 1 times 10 to the 9th base that is replicated, one of those bases will be a mutation, meaning the DNA polymerase makes a mistake 1 time in 10 to the 9th replicated bases. I believe that's 1 mistake in a billion nucleotides that get replicated. Since each gene has about 1,000 bases with it, that means the mutation rate per gene is about 1 in a million, or 1 in 10 to the 6 replicated genes will be mutated. Now, when we look at a human born in the human population is much greater than a million. In fact, we're close to, what is it? We're getting close to 8 billion? I used to know this number. I think we're getting close to 8 billion. So in the human population, whenever a baby is born, for 1 gene we look at, 1 in a million babies will be mutated in that gene. And like I said, the population of the humans is much greater than a million, and it's something like 8 billion. So we're getting humans born in the population almost every day where there will be a mutation in that 1 gene of interest to us. Any question about any of that? All right, mutations increase the mutation rate about 10 times to 1,000 times, and that increases the mutation rate in a replicated gene from about 1 in 1,000 genes to 1 in 10 to the 5th genes. 10 times 1 in 10 to the 6th is 1 times 10 to the 5th. All right, any question? That would be 10 times an increase in the mutation rate. You can test for mutants experimentally by positive direct selection or negative indirect selection. Positive direct selection is where you detect a mutant cell because they grow or appear differently. Like if this individual go walking down the street, everyone would say, yep, that's a mutant, because they look and grow differently. A negative or indirect selection is when you detect mutant cells because they do not grow. And let me explain this. I did this actually as an undergraduate in college, where I would take a plate and then replica stamp that plate with a velvet surface, and then take that velvet plate and plate it on 2 plates of agar. One of these agars has the amino acid histidine in it. The other does not have that amino acid in it. And then we go look for a mutation where there's a colony that does not grow in the media lacking the amino acid histidine, but it does grow in the media that does have the amino acid histidine. And so we know that these cells here, which I believe are these cells here, that this is a mutant for the amino acid histidine. And to get this mutant to grow, you have to supply it with the amino acid histidine. That is negative selection, which I actually did as an undergraduate. Any question about any of that? All right, let me see how we're doing on time. All right, I'm going to end the lesson here. Are there any questions? Oh, I got two here there. And we'll continue with this next time. We're a little bit behind schedule, but oh gosh, we have about 10 more slides to do, so we're a lot behind schedule. All right, I'm going to log out and say goodbye.